I would like to thank organizers for this great conference and for giving me opportunity to deliver this talk. First, I would like to start with disulfide bonds. So, disulfide bond is a covalent bond. It's a very strong one, and if you remember, it's 60 kcal per mole to break this bond. It's very important because in most of the simulations, we don't want to break this bond. It's not we are not able to break it unless we have oxidizing or reducting agent. So we'll not form it or reduce it unless the redox potential is included in our simulation. So in most of cases which we simulate theoretically, we don't care about disulfide bonds. They either are there as a harmonic potential or are as a Gay-Byrne potential or Lennard-Jones potential in all atom simulations when they are reduced. So it's not the case in vivo. In vivo, we have multiple number of enzymes, and we have a lot of oxidizing agents which react with disulfide bonds. I'm sorry, can I have the time on? Okay, great. So far, so good. So in vivo, we have the conditions which we don't care about in our simulations. So why disulfide bonds are important, and I would like to convince you that they are important, to take into consideration their breaking and their opening. In case of the protein stability, for instance, to the aging like keratins in hair or feathers, we do not care much about it. They are so stable and they are so isolated that they will not break so easily, or the time scale for their breaking is so long that we don't perform such a long simulations. But whenever we have the multimer studies without disulfide bonds for adiponectin, we can have with disulfide bond we can have a hexamer, but without disulfide bond we have a trimer, and this indicates different signaling path. So disulfide bond can be critical for many hormones. For instance, for oxytocin pressin ring, if we don't have this disulfide bond, it's inactive. Only after the pressin ring is the disulfide bond is present, the oxytocin will perform its function properly. So what is also the meaning of disulfide bond? It's also performed very important regulatory functions. One of the important function in our organism is that we don't want to oxidize too many things. And if we have oxidation stress. So oxyreductase is in most of the time in inactive form. It has disulfide bonds reduced. However, when there is oxidation stress, there is activation of transcription factor of oxidizing reductase. So it was proven directly in this publication that formation of disulfide bond is essential for the production of transcription of glutaredoxin 1. But once it is formed and we come back to the correct reduction environment in our body, or cell, sorry, then there's a deactivation of transcription factor of oxyreductase. So bearing that in mind, we have to remember that dynamic changes of disulfide bonds are essential for many protein functioning. So how frequently we should take care of disulfide bond? How abundant is disulfide bond in protein? We have about 100,000 structures, and 28,000 structures contain disulfide bonds. It's 23% of PDB structures deposited. I'm talking only without the ligand. So 23% of our simulations of proteins should involve breaking or formation of disulfide bonds. So in UNRES force field, how we treat it? UNRES force field was a little bit talked yesterday, mainly by Czarek. So we have two sides of interaction, one as side chain, second one as peptide group. So it will be about interaction between side chains. So what is different is from other force fields is that we can treat not only as a simple harmonic restraints, so that we say that they are always formed, or simple reduced one when we in UNRES force field we use Gay-Burn potential, but we can simulate formation and reduction simultaneously. So we have a fragment of potential which is similar to harmonic one. We have a potential which is Gay-Burn one, and we have in between transition state. And we can manipulate the potential of oxidation. We can manipulate the barrier of the height. But there is a problem with it. It's not perfect potential. One of the problems that all disulfide bonds are treated the same. And we know that it's not the true. Disulfide bond reduction potential strongly depends on the vicinity of this disulfide bond. And the frequency, how often this bond is formed, depends whether the enzyme is able to approach. It's not included. So disulfide bond has to be on surface to be able to enzyme to approach. Otherwise, it will not form, or the oxidative agent to approach. Another thing which is important is that it doesn't consider the vicinity of the disulfide bond. So we treat all disulfide bonds to be possible to form, and I would like to stress here, we don't a priori assume that which pairs should form and which not. All possibilities can be formed. So with this result, I would like to show about one HYP protein. And this is a protein with 4 disulfide bonds. I checked yesterday, and it's not a lasso protein. And it's not knotted. So all the conclusions, by the way, all the conclusions which I will make, it's not about lasso or knotted proteins. But important thing here is that this is signaling, lipid transfer protein, which is from plant. It's a very allergic one. So its shape causes allergy in human. And when we simulate with replica exchange molecular dynamics, which detail well covered by Czarek yesterday, we obtained it without disulfide bonds. We get the shape, the dominant cluster is of RMSD 6.6. So basically the shape is preserved, but the informational details are missing. But there are many, many possibilities to study this. So the most ideal situation is when we have all 4 bonds possible to form. So this is also the most complicated one. So this results in many traps, kinetic traps. But we simulated all these possibilities. And it turned out that when we have the 8 to 43 and 45 to 47, we get the best results in terms of the dominant cluster. When we have all disulfide bonds, it is more or less of the resolution which we obtained without. So I would like to stress that disulfide bonds are not the factor which drives us toward the native structure. They can create traps on our pathway, which will slow down the folding process. And it's not associated, as yesterday Joanna said, with threading through lasso. There's more into it. So when we look at those two, are the one which bind the ends, and they create the tertiary structure. However, when those, the second one which damages or impedes the one, are in the middle of the structure. So this is one of the downsides of this potential. So this one, they are not, this disulfide bond is not so exposed. So the frequency of formation and breaking should be different than those two. Nevertheless, we can draw the conclusion that it influenced the folding. And when we look how fast they are formed, those two which enhance the folding pathway are formed extremely fast, while those two which impede the folding pathway are the ones which are formed later on during the folding simulation. So let me go a little bit to the atomic force microscopy. It's experimental technique which we can get a lot of from. So we can get the binding energy of ligand and molecule, we can get the profiles energy, we can get cell elasticity, and many, many more. And there's a steered molecular dynamic. It's a technique that mimic atomic force microscopy, or try to mimic atomic force microscopy. And two most frequent variants is that we pull with a force from two ends, or we put a spring in between, and we push or pull with a constant velocity. I will be talking only about the second mode. This is a contacting domain of signaling protein. So what we can get from atomic force microscopy, this is about DNA. And I will advertise a little bit this article. So we have a telomeres. We studied telomeres, and we started to stretch telomeres. And we pulled telomeres from 5' ends. So, okay. My LibreOffice crashed. And I don't know why. Sorry. Okay, fine. I will just, which one? Okay, I'll try to be more careful next time. Let me just skip to the. Okay. So when we simulated the telomeres, we obtained that telomeres behave differently than any other sequence. And we find that telomere behavior was associated with the ability to wrap around. It has a unique ability to stabilize. No, there's definitely more into it than that. I'm terribly sorry. It crashed. Let me just go through. Yeah. So we also tried to no, my LibreOffice is getting nuts. I get I get the good screen here, but the bad screen there. I'll just copy the screen. Mirror. Apply. Fine. Okay, so when we simulated the telomeres, it turned out that there was one sequence, which I didn't show here, which could mimic telomere behavior. And there is a problem with plugin here. I'll just skip it. Go through. Okay. But I really advise to look into the paper. Okay, now it's working. Okay, now I'll shift with buttons. So we also performed with the 2EE2 PDB, and we found that we are able to reproduce the two experimentally predicted jumps, and the extension perfectly matched our results, while there were also all-atom simulations, and there by Mikulska and collaborators, the coarse-grain model, they obtained different extension results, but still two jumps. And when they analyzed what is the most stabilizing factor, they found that beta fragment is the most important one and the most stable one. So why we choose ribonucleases? Ribonucleases are as one of the most studied protein. It has a beautiful structure with a lot of helices, a lot of beta sheets, 4 disulfide bonds, and 2 prolines in cis configuration. And this is a very, very simplified landscape. Yesterday, Jose tried to convince us that the landscape of the protein in many cases is very simple, and that's true. But not in this case. Ribonucleases A, funnel for folding, is nightmare. It has, I'm only considering slow forming of pathways, possibles, and traps. And it results in 1,680 possible slow folded traps. By slow folded, I mean either disulfide bond formation or cis-trans of peptide group isomerization. So this is the perfect example to study influence of disulfide bonds. So when we studied influence of thermal stability of disulfide bonds, we found that depending which one is, there is a strong dependence where of the position and the disulfide bond. We found even that 2 disulfide bonds are extremely strongly formed, and I would like to keep in mind those two, 58, 110, and 65, 72, that throughout whole temperature range, we have preserved almost 100% of formed disulfide bonds, even in a high temperature. Whereas two other bonds, fraction, is significantly lower, especially 40 to 95. So keeping that in mind, we decided to do a steered molecular dynamics of RNase A. And we performed all-atom simulation with Amber 14, where we had Pauling constants of 14 kcal per mole with implicit solvent. There might be a question why implicit solvent. When you do steered molecular dynamics with explicit solvent, it would result in box of 500 angstrom. So we would have around 4 million water molecules. So we would mainly simulate water instead of our system. And it will slow down our simulation. It's already very slow when you perform steered molecular dynamics, but on GPU. But getting those results would be really nightmare in explicit solvent. So we also performed, for comparison, simulation with UNRES force field. And there's a velocity difference. So with all atom, we had 10 meters per second, which is absurd. It's non-realistic in any way. It's not possible that in vivo you have, basically it means that you would rip around a human body with that speed, not to say about the protein. So in UNRES force field, it's possible to slow down. And by meaning slow down, because I tried to mimic as fast as possible in UNRES force field, and still I have order of magnitude slower than in all-atom simulation. But this results millimeters per second. It's still way larger than in any experimental AFM. Here we have millimeter per second, while the fastest AFM, which was performed only because of to compare with all-atom simulation, were done with millimeter per second. And those were specially designed to be really fast. Normally, we pull with nanometer per second. This is typical order of magnitude. Sometimes slightly large. So we also decided to see how it is influenced when we have strong reducing environment. Weak oxidizing, and by weak oxidizing I mean that still disulfide bond is preferred one, but it's not very strong one. This is more or less to mimic in vivo condition when we look from the redox potential. And when we have strong oxidizing, what happens when we have oxidation stress? So first, let's compare Let's compare results from reduction when we have reductive environment. We performed all-atom simulation when we have reduced cysteines. So we get two jumps, a lot of fluctuations. The fluctuations come from that, bear in mind that AFM pooling is very sensitive from trajectory to trajectory. So to get meaningful results, you have to perform a lot of them. So this is from average from 10 simulation. While in UNRES force field, I performed 80 simulation. So this, just to compare the speed to obtain all-atom simulation on GPU, it takes 2 weeks, while with UNRES force field, it takes 6 hours. So this is a tremendous powerful tool in terms of coarse graining. And the position of the peaks really nicely match the position with all atom. And there is a huge difference in terms of forces obtained. But when you compare, when we slow down the pooling speed from 0.01 to 0.02, the force is decreasing. it's clearly the effect of pooling speed that protein resistance force is larger. Also, it is shifted to the larger values, which is also effect of pooling speed. The larger the pooling speed, the more shifted it will be to the larger C to N terminal distance. But this is known from literature. So we get the very nice comparison between the all-atom and UNRES results for AFM, so which more or less validate our results for reductive environment. So what happens when we compare with weak oxidizing with strong reductive? So now we have ability to form and break disulfide bonds, and look at those two plots. They are more or less identical. So in vivo, as it was cited in the paper of Zhang, most of the proteins are in reduced form, while even if it's formed, it doesn't significantly stabilize. But let's see a little bit further. So here I want to show what happens when we have oxidizing. And I performed simulation with either start from minimized structure with disulfide bonds formed or with them reduced. So I allowed them to be formed. So it doesn't matter on your simulation whether, because for instance, if you have weak oxidizing with starting from formed or unformed and reductive, all three look the same. When you look at the strong oxidizing and you have starting from not formed and formed disulfide bonds, they both look the same. So in AFM, it doesn't matter. It shows that starting configuration doesn't affect much, which is good news. But so it was able to form anyway disulfide bonds, even when we started from reduced one. So let's look what's going on in reductive environment. In reductive environment, we have two dominant peaks. And when we look at clustered structures around 240 extension, we see clearly the beta sheet at the end is the one that is preserved. So this is response for the first force jump here. And when we compare this obtained structures in UNRES, it's exactly the same. The C-terminal beta sheet is preserved one. And what news we get from it? That C-terminal beta sheet is extremely stable mechanically. Can we draw any conclusions from that? We know from experiment that if we cut 7 residues from C-terminal, ribonucleases say do not form anymore. So I'm not doing an overstatement, and I don't want to you to know that it's a conclusion, but the beta sheet, if you do perform AFM and you find something extremely stable, it might be a hint where to look for your folding seed. It not necessarily might be the true, but it is a good hint to look for it. And when we look at the second peak, when we look at the second peak, here it's more complicated picture because one of the beta sheets is banded here. It's not reflected. It's slightly different in UNRES. It's not reproduced. the dominant, but they more or less look the same. So that the stretch starts from N-terminal, and then the preserved is the beta sheet core. And now let's look at the story. What happens when we have oxidative environment? When we have oxidative environment, the first jump here corresponds to breaking of 2684 disulfide bond. So we get, in oxidative environment, additional mechanical stability. So RNAse becomes suddenly, when we have oxidative stress, very stable structure. And the one first to break is also from the N-terminal. And then the second peak corresponds to the breaking of 40 to 95. And as you remember, when we analyze stability from thermal one, those two were the one to first break. And the 6572 and 11058 were the one the most stable. And we have the same results here. So it clearly indicates that those two disulfide bonds are the one extremely stable. So when we look also on the folding pathway, this might be indicative where to look for which disulfide bond is formed first. And there's another tremendous peak here, which is way, way, way larger in here. And what happened? So this is very similar to what we previously see. We have beta sheet structure preserved, which is similar to the beta sheet which we had previously for this one. But look at one detail. Here, we have a new disulfide bond formed between non-natively existing disulfide bond. So there's additional stability that comes that during the, if something wrong happens, there is a catch that it still can form the wrong pathway, which we would like not to be there. Because we have then a beta sheet which would easily aggregate, which is extremely stable. So going to the conclusions, I would like you to keep in mind the beta fragments of RNases are the most stable one. So that experimentally found the beta sheets is the one from C-terminal to forms first is also confirmed here. And disulfide bonds significantly stabilize RNase A. And the stabilization is oxidation potential dependent. And bear in mind that it's extremely relevant biologically, as in oxidative stress, we want to keep our protein extremely stable. And I would like to thank collaborators with whom I performed this study, and National Science Center of Poland for the funding this study. And I would like to thank you for attention.